I am going to be talking very briefly about our recently published work in the Journal of Proteome Research. This work is an highly collaborative project. It's highly collaborative with input from various labs. The paper is from the Fernandez lab from Georgia Tech. I am a co-first author on the paper, and I actually was in charge of the computational aspect of this work. Samyukta Shah is also a co-first author, and she was the one that was actually in charge of the mass spec aspect of this work. So this is going to be a very brief video abstracts, just walking through some of the main figures in the paper. So let's start with the abstracts that was in the paper. Ovarian cancer is one of the deadliest cancer that's affecting the female reproductive system. And high-grade serous ovarian cancer is a subtype that's responsible for the most cancer deaths, and that's the subtype that's actually investigated in this work. Little is known about the metabolic cause of this disease. So how does the metabolites, how does the lipids, if you will, changes as the ovarian cancer progresses? And in this longitudinal study, we actually examined the temporal cause of the serum lipidome changes using an high-grade serous ovarian cancer, what I would call the HGSC mouse model, and using machine learning. And we were able to uncover quite an interesting dynamics of the remodeling of the lipids that's actually happening as the ovarian cancer progresses in this mouse model. So let's get it started. The study design and the blood sampling essentially is illustrated in this figure. We have the DKO mice. So the DKO is the mouse model for this ovarian cancer. HGSC, that's the high-grade serous ovarian cancer. It's a disulpidem mice, and it's just called DKO for short. And then we have a DKO control animal. So starting for 2 months, blood samples are collected every 2 weeks, and we have lots. Again, this is a longitudinal study, and we have lots of blood samples to work with, and that is so. After the collection of the blood sample, we did untargeted lipidomics using mass spec, and panel B essentially is showing the way we are actually doing the comparison. And we took all of these granular blood samples that are actually collected in order to make the DKO mice comparable to the DKO control mice. We actually converted into something called a percentage lifetime stages. This is just to make them more comparable, and we have about 5 lifetime stages for these mice. So from 0 to 30% lifetime stages, that's going to be lifetime stage 1. From 30 to 45% lifetime stages, that's going to be lifetime stage 2, and so on, as indicated. And after that, we actually did a bunch of computational analysis to really uncover what is the pattern of what's going on. So global lipidome changes, longitudinal lipidome evolution, ovarian cancer lipidome signature, and of course I did actually some survivor analysis. So let's get this going. I'm just going to be walking through the main figures very briefly. So panel A here is showing the global lipidome changes. This is the overall fold changes for all the annotated lipids that was annotated in this study, and this is for all the time points combined. As you can see, there's so many perturbations going on. And for panel B, we're essentially showing a subset of the content from panel A, and this is just 87 significant lipids that were detected, and this is essentially showing those perturbations. Again, on the y-axis, we are seeing this is a log2 fold change of the DKO relative abundance to the control relative abundance. Panel C is the number of significant lipids over time. That's the lifetime percentages. You can see that from 1 to stage 5, and it largely increases as the ovarian cancer progresses. Panel D is the offset plot that's showing the overlapping significant lipids in various lifetime stages. And these result from this figure would actually come into place when I'll be discussing the prognostic circulating lipid candidates. So, lipidome changes that we see in response to ovarian cancer progression. What I did in this aspect of the work is to actually compute something called the hierarchical clustering analysis. So I took all of the lipids, and I essentially try to just in an unsupervised manner cluster them and see what emergent trends do we have, and we actually ended up coming up with about 4 biologically meaningful trends, and this is the cluster A, B, C, and D. And in cluster A, we mostly have phosphatidylcholine's, phosphatidylcholine ethanoamides, and these are mostly phospholipids. And those clusters, those lipids in those clusters are quite similar to what we see in cluster B, cluster C. And it appears that the overlapping trend here is this uptick that we see from lifetime stage 1 to lifetime stage 2, right? As you can see, A and B and C. For cluster D, they consist mostly of single lipids, so exoceramides and ceramides, and that is also distinctive of this uptick towards the end of the lifetime of these mice. Okay, and again, the y-axis here is comparison between the DQ and the control, and panel C is just showing a network graph of the lipids present in each of those 4 clusters. We did machine learning to discriminate between the DQ and the DQ control mice. So we have random forests. Feature selection was used on the training data sets, and we selected features that have Gini index that is greater than the mean of the Gini indexes. Then we train with those subsets on the training data sets. We then predict class membership using the test sets, and 5 machine learning methods we use: logistic regression, random forest, k-nearest neighbor, support vector machine. And I also added a voting classifier, and this voting classifier essentially incorporates, it's an ensemble method of consisting of these 4 previously mentioned machine learning methods, and soft voting was used. And each of these was applied to each of the 5 lifetime stages to actually do the training, to make the test prediction. And additionally, we did some validation of the classifier models. So these are the best performing classifier model with permutation test. So in summary, in B, C, D, E, and F, I'm just showing the ROC AUC test score for all of the machine learning models for all of the lifetime stages, as you can see. And panel G summarizes these results, just indicating the highest test score ROC AUC values for each of those lifetime stages. And quite auspicious is the fact that even at stage 1, we are seeing a 0.8 AUC values, which is actually quite interesting, and actually erodes the promise of an early stage markers if we have similar lipids showing up in humans. Okay, so these are the discriminant lipids. That's from the machine learning analysis that were found in each of these lifetime stages. I won't go into too much detail. We discussed this extensively in our paper. And at the early stages, we see mostly phospholipids. You can see that in A. So this is the lifetime stage 1, lifetime stage 2. But as we go increasingly further, we see a much mixed bag or mixed alterations in terms of lipid classes. And these are the details of those lipids that were actually identified as discriminant lipids. Okay, so this is one of the last analysis I did, and this is just a prognostic circulating lipid candidates, presenting them. And essentially, this panel A, B, C, and D are showing a volcano plots that actually compares just the DKO now, because essentially we are looking for prognostic circulating lipid candidates amongst the DKO, of course. So in panel L, we are comparing the DKO lifetime stage 2 compared to 1, DKO lifetime stage 3 compared to 1, DKO lifetime stage 4, and so on and so forth. And the significant lipids that were actually selected here were actually presented in this offset plot. So this just basically showing the unique numbers, the number of overlaps, and the colored overlaps in brown, essentially showing the lipids that are present. Okay, the significant lipids are present in at least 3 of these 4 comparisons, and these lipids were actually used to screen for prognostic circulating lipid candidates, in addition to some lipids from figure 1 that are presented earlier. And to do that, essentially we just did what's called the median splits. We take all of a DKO group for those lipid candidates, of course. So we have a DKO low group. These were built for lipids with lipid abundances lower or equal to the median of the relative abundances for the selected lipids. Why the mice, and those are the DKO low group, if you will, and why the mice with the abundance greater than the median are actually bound to give you the DKO high group. And this is just showing the Kaplan-Meier curve comparing the low group and the high group for each of these phospholipid phosphatidylcholine species. And this is the Kaplan-Meier curve, and then you can then see using the log-RAC test, the p-value are actually quite significant. And these are the prognostic circulating lipids that we presented in the paper. And in figure I, we're just showing how does that lipid actually compare between the DKO and the control. So it actually is at higher levels in the DKO, in the DKO mice. So in the conclusion in our paper, what we did essentially is to then show a schematic displaying the key metabolic alterations that we actually see from our analysis, and we leveraged on our cluster analysis. So what lipids are detected in those clusters that I presented earlier. We also leveraged on the discriminant lipids that were detected using machine learning. And we used that to essentially build this visualization of what we think might be going on. And we're of course seeing perturbations in triglyceride synthesis. We're seeing perturbations in the Kennedy pathway, in the Land cycle, ether lipid metabolism, fatty acid synthesis, and so on and so forth. And these are actually quite discussed in detail in our paper. But in conclusion, we did a deep temporal lipidomic study of ovarian cancer mouse model. Phospholipids were the most perturbed lipid class, and they are also represented as the highest number of altered species at the early stages of this ovarian cancer progression. And perhaps this is rare because of the idea of the cell integrity fortification, because we know phospholipid actually is an important part of that cellular process. At the later stages, we found more of perturbations in the sphingolipid pathway. So ceramides, exoceramides, were predominantly increased in the DKO mice, and also several other lipids actually. Anyways, this is just meant to be a short summary. You can pick up our paper for more detail, and thank you for listening.